Hey guys, what's up? We're gonna finish up the kidney and talk a little bit more about the urinary system. So where we ended up last time was discussing the pressure in the glomerulus and the factors that affect that, leading to a net filtration rate of about 10. Um, as we progress through, obviously that was more or less the first part right here. And we talked about that filtration and things that should go through and should not go through. Um, good. Now we'll work ourselves through here. I like this picture because it kind of just summarizes everything. We could see the PCT, right, proximal convoluted tubule, and we can see that it is the primary site for reabsorption. Ions, water, various nutrients like glucose and amino acids that we'll talk about, and then we'll also talk about how they're transported. Um, as we move down, you will see that you have if this thing draws, descending loop of Henle. Um, and there are aquaporins located here, and we're going to talk about them a few times today. Um, and what an aquaporin is a protein channel that allows water to move through, right? So the descending loop is permeable to water, and it allows water to move from that filtrate into the interstitial fluid surrounding that loop. We know that the loops of Henle can be different lengths, um, depending on how far they're going down into the medulla. Hence, we have the two different types of nephrons that we discussed previously. And there is a difference from the descending loop, which is permeable to water, and the ascending loop that is not, right? So you can see that they called it the thick loop. It's a little bit thicker. It does not have those aquaporins and it is not permeable to water. So what happens here is we have the reabsorption of sodium and chloride from the filtrate into the interstitial fluid. So we can think of this sodium as kind of piling up in here, making a very salty environment, which aids, right? We know water wants to move towards more stuff. So that aids in the ability of that water getting pulled out into the interstitial fluid. And this whole process here we call kind of the counter current mechanism. With the water coming out, salt getting reabsorbed, et cetera, allowing for concentration of urine. So a little bit of water reabsorption taking place in that loop to further concentrate the urine, which is extremely important. Over here by the distal convoluted tubule, the DCT, right? You can see that that is after that ascending loop. It's after the loop of Henle. Um, and primarily what we talk about here is secretion and regulation of certain ions to help maintain the pH and the electrolyte balance of the body and the filtrate, right? So it's kind of where we have a little bit of secretion that happens elsewhere, but this is really what we talk about with the secretion in the DCT and the collecting duct. And what the main role of the collecting duct is, and we'll talk about hormones that work in these various areas, etc., is reabsorption of water. I like to think of the collecting duct as the last site, right? Because whatever comes out the bottom of that collecting duct is going to go through those entire pathways we talked about, right? Collecting ducts to the minor calyx, to the major, to the renal pelvis, to the ureter, to the bladder. So this is kind of the last site for water reabsorption. So it's very important if we are trying to keep water, and there are various hormones that work on this area, that collecting duct's able to reabsorb it. And we'll kind of go through some details with that as we progress forward. We got a video. Check it out. Kind of helps you out with all the things that we've been talking about. But what we're going to talk about now um, is the control, right, of how the kidney can regulate the GFR or the glomerular filtration rate, right? There are three mechanisms in general. There's renal autoregulation, there's neural regulation, and then we have hormones that help regulate, and we'll dive into each one of these. The main thing that we're talking about is, as we talked previous lecture, stuff gets filtered in the glomerular capillaries, right? So the big picture is stuff's getting filtered through here. We talked about those pressures, etc. But it is dependent on blood flow coming in to the afferent arteriole and leaving through the efferent arteriole. So you could think of these as two little blood vessels that can open and close independently. And the way I like to think about it is the more blood that comes in, the more filtration we'll have. So as we'll see some pictures and I'll kind of explain it, these are the factors. So what things affect how dilated the afferent arteriole is or the efferent respectively will change how much blood in general is in here, and thus it will change how much blood gets filtered. Because if the blood's there, it's getting filtered. So the only way you can control it is change the amount of blood flow coming in or out. Hopefully that makes sense because we're going to talk about it. So here we have what I was just talking about. We have the afferent arteriole, and you can see here it's constricted. So what does that do? That reduces the amount of blood flow, and this kind of just, it's a simplified version of the previous picture. Right? This middle section's our capillaries right here. Right? So you can think of that filtration occurring here, if that helps, based off how much blood flow is coming in. So if we constrict that afferent arteriole, we will see less blood goes in and the glomerular filtration rate decreases. Pretty straightforward. If this is not doing anything, let's say it's dilated or opened, and we constrict the efferent arteriole, blood is coming in, but it has a hard time leaving, so it stays longer in that capillary bed, and thus, do low, and thus that hydrostatic pressure increases and more stuff gets filtered because there's more blood sitting in there. So basically you close, think of it like if you're taking a shower and you have a lot of hair buildup, right? In the drain, the water's going to back up, right? So it'll start filling up. That's the same thing that you can see here. And thus, because that exit is closed a little bit, the blood stays in there longer and that glomerular filtration rate increases. Now, what factors control this? Right? There is a very important area of the nephron. And in reality, like when I show you the nephron, we like stretch it out and we draw it kind of in a pathway. Like the reality of it is it's flipped on itself and they're all crammed into very small spaces. So the ascending loop, where this comes up and turns into the DCT. It's like the end of the ascending loop, and on the further slides, we'll say the beginning of the DCT or the DCT. You can see that it's located right next to the afferent arteriole. So the next slide that we're going to look at shows this zoomed in, and we'll talk about two groups of cells that are extremely important. So you can see that it makes contact with that afferent arteriole. And there are specialized cells that are located here that make up a functional group called the JG apparatus or the juxtaglomerular apparatus. And this group is responsible for regulating that first component that we talked about, renal autoregulation. So it is in the kidney, obviously, they're nephrons. And depending on what's going on in this region, it can self-regulate how much blood it's filtering. And that's the big picture, and it's significant. So when we zoom in and we look at this here, it's just a cross section, right, of the previous slide that we were looking at. See if I go back, boom, right there. You can see that this afferent arteriole has specialized cells. These are called juxtaglomerular cells or JG cells. Right next to that, you have macula densa, which are specialized cells found towards that distal convoluted tubule, right where that ascending loop goes in. These play a very, very important role. Look at the proximity to this afferent arteriole that we were talking about how it regulates the amount of blood coming in and thus regulating filtration. So, take a look at this. There are a few different components to this. It all references this area, right? First one is we have myogenic mechanisms to adjust the GFR. As that afferent arteriole stretches out, right? Stretching causes constriction. So as it stretches, you can think of it almost like a reflex. It constricts. As that afferent arteriole constricts, reduces blood flow, reduces blood hydrostatic pressure, thus less blood's coming in, and it will overall reduce the glomerular filtration rate. We call that the myogenic mechanism. The second mechanism is called tubuloglomerular feedback, and it's relatively similar. But cells in the DCT, the other cells, can detect the rate and what is in the filtrate, right? So if it's filtering a lot or filtering a little bit, it can detect that. So if filtration is too high, so too much stuff going through there, you don't want to lose everything. It inhibits the release of nitric oxide, which is a vasodilator. And if it inhibits that vasodilation of that afferent arteriole, it will constrict and thus reduce the amount of blood flow in, right? So the myogenic deals with the muscle of the afferent arteriole. The tubuloglomerular feedback deals with the cells of the DCT that can kind of detect how much filtrate is being formed. And then we have a neural regulation, right? But again, that was kidney regulating itself. So and we've talked about this because we've talked about the autonomic nervous system extensively. But that afferent arteriole is innervated by sympathetic nerve fibers. Duh. So when the sympathetic nervous system is activated, aka the ANS that we are very familiar with, hopefully, it leads to release of norepinephrine and it causes vasoconstriction. So under sympathetic stimulation, right, we can see that this thing kind of comes down here, right? Under sympathetic stimulation, we're going to constrict that, reducing blood flow in and thus reducing glomerular filtration rate. So although there's different causes for all of this stuff, the big picture is how do you change the glomerular filtration rate? When do you want it to be higher? When do you want it to be lower, right? Obviously, during sympathetic stimulation, probably don't want to be filtering out a tremendous amount of blood. You need that blood. The body's quite busy doing other stuff. Thus, we decrease the glomerular filtration rate. And lastly, and a very important one that we also talked about before, is hormonal regulation. So if we recall, maybe, you have angiotensin 2, which was part of that renin-angiotensin-aldosterone pathway. We talked about it when we did the endocrine system, and we talked about aldosterone and what it does. But what angiotensin 2 is, right? If you remember, you had renin, angiotensin 1, angiotensin 2, aldosterone, that whole pathway. It is a very, very strong vasoconstrictor, right? Constrict that afferent arteriole and the efferent arteriole, less blood comes in, and you reduce glomerular filtration rate. Makes sense, right? Why would we do that? Because we want to save that water, and the goal is to increase the blood pressure because that's what that pathway is involved in. Check. And a weird hormone, and it's only weird because everything else we've talked about so far was designed to reduce glomerular filtration rate. ANP, atrial natriuretic peptide, comes from the heart, right? The atrium of the heart can release this. When you have high blood volume, so think about that. Everything else is designed to kind of detect low blood volume and increase, save water, save ions, etc. This one is released during high blood volume. So what it wants to do is make those glomeruli, those capillaries, more permeable. It increases the surface area, and it increases the glomerular filtration rate. This is one of the only things that can actually help you lower blood pressure, right? By increasing glomerular filtration rate and getting rid of more liquid. This is a very important chart that I just described to you, but it's a nice thing to kind of go through and familiarize yourself with. And as stated, the effect on glomerular filtration rate, all of them cause a decrease except for the ANP. And it talks about how, obviously, all of this stuff is pretty much narrowing the lumen of the afferent arteriole. So slight differences as far as the stimulus, but the overall effect is pretty much the same. What things get reabsorbed along this entire process? We pretty much want water, proteins, sodium, chloride, bicarbonate, glucose, too many, and potassium to stay in the body. We need that stuff, right? So you will see that the majority of this is reabsorbed. However, two components in general are secreted, and that would be our urea, uric acid, and creatinine, right? Uric acid, it's leaving, right? It doesn't necessarily more secreted, but it's in there. So you could see what things we want to save. If we are saving it, we should not see high amounts of this in the urine, right? And we'll talk a little bit about urinalysis, but we should not see high amounts of this in the urine. Obviously, in lab, you go through urinalysis as well, and I tell you how to do it, etc. So kind of as a summary, right? Secretion and reabsorption. We want to save these things, and we can get rid of these things. Pretty straightforward, right? This is another kind of drawing that summarizes a lot of things, but it goes through and it talks about what the um, major kind of functions are of each of these parts, and then it gets into hormones, right? And where they work specifically on the nephron. So we know the PCT is the site where most solutes are reabsorbed. It is the primary site for water reabsorption. 65% of water is reabsorbed in this area. We've talked about it. And the DCT and the collecting duct are two areas besides the glomerular regulation where our hormones primarily work. This is important. This is the end of the nephron, and this is where we could really influence what we're saving or what we're getting rid of. Now, this slide is talking, it doesn't say it, but it's talking about the collecting duct, right? As well as perhaps a little of the DCT. Lining the tube of the collecting duct, we have things called principal cells. These cells are designed to respond to the hormone ADH. We've talked about ADH before. Synthesized in the hypothalamus, stored in the posterior pituitary, gets released, and it allows us to save water. Anti-diuretic. A diuretic makes you pee. It's the opposite. So what does this do? The ADH makes us add aquaporins to this membrane. So this is inside the nephron. There's water there. This is the last site. We want to save the water. So what it does is it takes these few aquaporins and it allows us to add more, and just straight up, we suck up more water, which means that we save more water into the blood, and we concentrate our urine. So key thing here, principal cells found in the collecting duct respond to ADH, and in so doing, increased aquaporin synthesis and attachment, and it allows us to reabsorb more water before it exits out into the blah, blah, blah sequence, ending up in the bladder. So some hormones that we've discussed already. ADH, right, works on the DCT and the collecting duct. Aldosterone works on the DCT and the collecting duct. So it's kind of scribbly right there, but it's all good. So you could see these and these both work on the DCT and the collecting duct because those are the last sites of reabsorption. Check. ADH primarily is focusing on water reabsorption. Aldosterone, as we've talked about previously, causes sodium retention, which then indirectly causes water retention, right? PTH, we talked about probably in hormones, is a hormone that helps us raise blood calcium. So it works on the DCT, and that would help us save calcium so it doesn't come out, so it can raise our blood calcium levels. So few hormones there work on the DCT. And on the collecting duct, obviously we have ADH, aldosterone, and ANP. And again, the goal with ANP is to get rid of stuff. So it actually it works opposite of the other two. You could see here, anything green is causing reabsorption, anything red is causing secretion. So it helps the secretion and the getting rid of the water and the sodium, right? Respectively so. We should totally be familiar with what's being taken in each of these respective areas, right? So clearly we've talked about this one a lot, and you can add to that 65% water, obviously. And here, this goes through the mechanism of how things are reabsorbed. Up to this point, I just said things get reabsorbed. Cool. Well, there's a way that things get reabsorbed, right? And we'll mention a few. And this goes back to A&P1, first or second lecture when we talked about how things are transported, right? Very small molecules like ions, sodium, potassium, hydrogen ions, can diffuse directly out of this key thing here. This is inside the nephron. This is our filtrate, right? And then things get reabsorbed into the blood. We're familiar with that. So things can diffuse. Some things can be actively transported, aka basically still transported relatively simply. It just costs a little bit of energy. The next way is a symporter. So this is one of the ways that primarily that glucose is reabsorbed, and it's important to understand this. And a symporter transports two things in the same direction. So you need two salt and one glucose, and then it gets shipped in. This is important, right? So the sodium symporters, think about it, sodium's being moved in, and these guys are just hitching a ride. Pretty straightforward. But it's very, very important. We need to get this glucose and amino acids back in, right? To the blood. They are too large to diffuse. Obviously, glucose is a little bit bigger, right? Amino acids are a little bit bigger. So this process of channeling this in for reabsorption is extremely important. Sodium, glucose, symporter. And things get secreted, right? Oftentimes via something called an antiporter. And an antiporter transports two things in different directions. So if we want to secrete something, it's almost like you have to exchange it. So here you can see salt coming in and hydrogen ions, aka acidity, being secreted. This is the first time we see something going from our cells or our blood back into the filtrate of the tubules. That happens via an antiporter. Sodium comes in, H plus gets secreted. Pretty straightforward, right? Keep saying that, ish. Sodium reabsorbed, hydrogen ions secreted. These are called antiporters. This just further reiterates, right? This is the thing that we looked at before with the angiotensin, the aldosterone, the ADH, the ANP, and parathyroid hormone. It tells you what the effects are. And now that we kind of understand how things are transported, when we see increases reabsorption of sodium in water, we have some idea of how that happens, right? It's either going to be diffusion or active transport. Increased secretion of potassium. We know that if we're going to secrete something, usually the things are going to go in opposite directions. So we secrete the potassium and we reabsorb the sodium via antiporters. Pretty much makes sense. So it helps us give a little bit more context to what we're learning. And it tells you the stimulus, obviously. And it tells you where in the nephron this happens, or we should say where it works, right? And you'll see a lot of this stuff is DCT, or the collecting duct, the principal cells. Makes sense. And now we get to urinalysis. So we talk about this a lot in lab. I go through the whole thing and how it is and what things we're looking for and specific numbers. There are a couple different types of tests. We have BUN tests, blood urea nitrogen, plasma creatinine tests, right? These are things in the blood. Creatinine clearance, and a few diseases associated. I'll probably put this in the study guide. You guys could kind of just be relatively familiar with it. But if you understand the anatomy, the question's which part is broken, right? And the last component-ish um, is the bladder, right? So we talk about this entire lecture how things work, how blood goes in, gets filtered, urine gets produced, clean blood comes out, right? That's what we've been talking about. So now that we know that we have this urine produced, hopefully concentrated and with good stuff in it, it travels down the ureters and fills up the bladder. This happens via gravity, peristalsis, and pressure of buildup of urine, right? So for the most part, we're not staying upside down all day, so gravity helps us drip this produced urine down into the bladder. Um, Clearly, if you have some type of obstruction, and we'll see on the next slide something that can happen, you have a backup of urine, and that could cause serious problems, right? Um, it could damage those glomeruli, right? Those glomerular capillaries. It could cause damage to the nephrons. It's clearly going to reduce your glomerular filtration rate. Um, and that's clearly no good, right? Um, so we look at nephritosis. Basically, you see here we have a um, inferior displacement of the kidney. Basically, it's floating kidney. So instead of it being attached and adhere to the specific wall and the structures we talked about last lecture, it kind of distends, and it can cause a kink in the ureter, which could result in that same problem that we were talking about with obstruction of urine getting to the bladder, right? Could cause serious tissue damage. And the bladder. So the way that we regulate urine output, um, simply, one of these components we have voluntary control over. We know for the most part, unless there's a problem, you can choose when to pee or not pee, right? It's kind of learned, right? Some younger kid can't do that yet. So in the bladder, you have rugae, which are these folds. We'll also see them in the stomach that allow kind of for that bladder. Think of it just like it's like kind of sprinkled up. And then as it fills up, it allows for the expansion and the stretching of that bladder. And we'll talk about how much urine this can hold. Um, you have specific muscles within the bladder that, when activated, will contract and force this urine out. And you have two sphincters. You have an internal, which is involuntary, which means you cannot control it. And then you have an external urethral sphincter that is voluntary. That's the one you can control. So once you kind of relax that, then the urine gets void. Um, this is looking at the bladder filling, first sensation to urinate, etc. So we could see here that the bladder could hold a little bit less than a liter, right? Depending on the individual, smaller or larger bladders, about 0.8 liters, right? But that sensation to where you really don't want to get maxed out all the time. That sensation to pee is about halfway filled, right? So we could see about 0.2 to 0.4 liters, 400 mLs ish, kind of starts to trigger that response. But again, you hold it, you go about your business until you can find a bathroom. Um, and the only reason you can do so is that external sphincter, which we have voluntary control over. So you could just be like, no, no, right? I think we see that on the next slide or two. Contract that muscle, no urine's coming out. Yep, good to go. And this just talks about some common problems associated with this, right? Leakage. So basically, you have that pelvic floor muscles are relaxing. So instead of keeping it in, right? Obviously, some of those pelvic floors, and there's certain times when we see this post-pregnancy and stuff like that. But some urine can drip out because the pelvic floor muscles are not contracting, or maybe they're weaker or whatever, and it can lead to that kind of leakage. And you have involuntary bladder muscle contractions that create kind of the consistent urge to have to pee, even though that bladder's not filled, right? So the bladder can contract. You can think of from the top down, but it's really not. It's like this, and it builds up that pressure, triggering those stretch receptors, making you feel like you have to pee. So voiding, urinating, etc. Um, we talked about there's a certain kind of threshold that will start to trigger this response that lets you know it's time to void the urine because you can't see it. And this is regulated by stretch receptors, right, which we could see here. And then they send messages, obviously, when those receptors, just like any type of sensory input, to the spinal cord. And then we have a nervous system response. So we like to think of it as three separate things. Well, they're all working together. Um, Parasympathetic one is going to be controlling this, right? So it's going to tell you to contract the muscles of the bladder. Parasympathetic, rest and digest, aka also pee. Um, the parasympathetic is going to inhibit, right, that internal sphincter. So it's not contracting. So now you have muscles of the bladder contracting, the sphincter, aka the gate, opens up. Um, and then obviously you control um, that lower sphincter, right? That external one. So once you inhibit that contraction, it opens up, and the urine comes out and you void it, right? So clearly, as stated, you can control that motor neuron influence over that voluntary sphincter. And this is just showing you what it is, right? Obviously, higher CNS input comes from your brain and your voluntary control, and that tells that muscle to contract so you don't pee in your pants. And then this is just showing you that parasympathetic. Oh no, it's just showing you the stretch receptors, and then what that leads to, right? So bladder's clearly full in this picture. Stretch receptors, complete distension of that bladder, send that sensory message in. Parasympathetic neurons cause contraction, um, and pretty straightforward. And we'll wrap it up, right? We have two different urethras in a male and female. We've discussed this before in the reproductive as well. The urethra is way longer in a male, and it serves bifunction, right? Not only is it responsible for the urinary system, but also the reproductive system, as far as sperm and semen released during sexual reproduction. And the urethra also passes through the prostate gland, which can cause problems as we age if you have prostate issues, like we discussed. And you got the gist of it. So that wraps it up for the urinary system. We discussed the basic anatomy. We discussed all the parts of a nephron, how the nephron works, what things we want to save or secrete, and thus what things should be in the urine. And we discussed a bunch of other stuff. And your next lecture will be on the digestive system. See you then.